Hi students, myself Satish Kumar Kundila, Assistant Professor, Department of Pharmaceutical Sciences, Vignan's Foundation for Science, Technology and Research, Guntur. In previous video, we discussed about the ketogenesis, how the ketone bodies are produced in our biological system. That is already discussed in the previous video. In this video, we are going to discuss about the biosynthesis of fatty acids. So we know that the dietary carbohydrates and amino acids, when they are consumed in excess amount, they can be converted into fatty acids, and after converting into the fatty acids, they are stored as the triglycerols. Okay, so during this process, how the carbohydrates, dieties or amino acids are converted into fatty acids in our biological system, we are going to discuss in this video. So the process which is occurred during the conversion of these carbohydrates into the fatty acids are nothing but the de novo synthesis process. De novo means new synthesis of fatty acids. So the process is described in this video. So this de novo synthesis of fatty acids occurs predominantly in the liver. The process of producing the fatty acids from the carbohydrates or amino acids is occurred in the liver, kidney, adipose tissue and lactating mammillary glands. So these are the locations where the fatty acid biosynthesis takes place. Okay, the areas are nothing but liver, kidney, adipose tissue and lactating mammillary glands. And the enzyme machinery or group of enzymes which are required for the biosynthesis of fatty acid production is located in the cytosomal fraction of the cell. So we know that there are different locations like liver, kidney, adipose tissue, lactating mammillary glands. So these organ cells, in that cells also cytosomal fraction has the enzymes which are required for the production of fatty acid from the carbohydrate or amino acids. Okay, and for the production of fatty acid, acetyl-CoA is the prerequisite. The acetyl-CoA is prerequisite for the fatty acid production. The acetyl-CoA is the source of carbon atoms, while NADPH. So this is also one of the requirement for production of fatty acid. Acetyl-CoA and NADPH are the two important starting compounds that are required for the production of or biosynthesis of fatty acids. So in that too, acetyl-CoA is the source of carbon atom and NADPH provides the reducing equivalents and also ATP supplies the energy for the fatty acid formation. Okay, and here the biosynthesis of fatty acid can be explained in three stages or three steps. First one is a production of acetyl-CoA and NADPH. The first stage is nothing but production of acetyl-CoA and NADPH. Second one is a conversion of acetyl-CoA to malonyl-CoA. Conversion of acetyl-CoA. So in first step, we are there is a production of acetyl-CoA and NADPH. So in the second step, the acetyl-CoA which is produced in the first step will be converted to malonyl-CoA. And last step is reaction of fatty acid synthase complex. Okay, there are the different sequence of events that will be occurred during the production of fatty acid synthesis. So let us see one by one. So first stage is nothing but, or first step of the biosynthesis of fatty acid is nothing but production of acetyl-CoA and NADPH. The acetyl-CoA and NADPH are prerequisite of the fatty acid synthesis. We already know that acetyl-CoA is produced in the mitochondria by the oxidation of pyruvate and fatty acids or degradation of carbon skeleton of the certain amino acids and from the ketone bodies. So these are the sources of acetyl-CoA. So the acetyl-CoA is produced in the mitochondria by the oxidation of either pyruvate and fatty acids or it may be produced from the degradation of carbon skeleton of the certain amino acids, or it may be produced from the ketone bodies. So these are the source of acetyl-CoA production in the mitochondria by the oxidation. And this mitochondria, so it is not permeable for the acetyl-CoA. So it is produced, the acetyl-CoA is produced in the mitochondria. So consider this one as the mitochondria. In that, acetyl-CoA is produced. Acetyl-CoA is produced, but it is not permeable. It is not permeable. The membrane of the mitochondria is not permeable for the acetyl-CoA. So that means it is not transferring the membrane of mitochondria. So we know that the enzyme system which is required for production or biosynthesis of fatty acid is occurred in the cytosol, of the cell. Cytosol of the cell. So the acetyl-CoA should be available in the cytosol, then only the enzymes will act on the acetyl-CoA to convert into the fatty acid. So for that reason, the acetyl-CoA should be transferred from inside the mitochondria to the cytosol. So for that purpose, there is a one phenomena, that is nothing but citrate. It will be the acetyl-CoA will be converted into citrate when the acetyl-CoA is condensed with the oxaloacetate. Oxaloacetate that is also present in the mitochondria, which is also produced from the pyruvate, and acetyl-CoA is produced from the pyruvate, and also this oxaloacetate is also produced from the pyruvate by the two different pathways, and these two combine together. That means they are subjected to condensation and they can be converted into citrate. And this citrate, so when the acetyl-CoA is converted into citrate inside the mitochondria, then it will be easily transferred to cytosol. It is easily transferred to cytosol. Okay, and this citrate, which is freely transferred into the cytosol, and then it will be cleaved. That means that it will be subjected to breakdown, and they can and it can liberate the acetyl-CoA and oxaloacetate again. So here inside the mitochondria, oxaloacetate and acetyl-CoA condense to form the citrate, and this citrate has the capability to cross this membrane of mitochondria, and then it will enter into the cytosol. There is a lyase enzyme is present to lyse the citrate. So thereby it can again liberate the acetyl-CoA and oxaloacetate in the cytosol, and this oxaloacetate in the cytosol is converted to malate. The oxaloacetate which is enter into the cytosol, that will be converted into malate. So here we can see the pictorial representation of first stage of the biosynthesis of fatty acid, that is nothing but the production of acetyl-CoA and oxaloacetate, NADPH. So inside the mitochondria, this one is the mitochondria. Inside the mitochondria, the pyruvate is converted into acetyl-CoA and oxaloacetate. So the pyruvate dehydrogenase will convert the pyruvate to acetyl-CoA, and pyruvate carboxylase is the one of the enzyme that will convert the pyruvate to oxaloacetate. And these two molecules, so why? Because the acetyl-CoA is impermeable to this through the membrane of the mitochondria. So for that reason, to enter this acetyl-CoA into the cytosol, it will be condensed with the oxaloacetate by the enzyme, that is a citrate synthase. So thereby these two, acetyl-CoA and oxaloacetate, will combine to form the citrate, and this citrate will easily cross this mitochondrial membrane and it will enter into the cytosol. When it will enter into the cytosol, there is a enzyme system, that is a citrate lyase. So it will break this citrate, or it will lyse the citrate to form the again acetyl-CoA and oxaloacetate. So this one is a acetyl-CoA and oxaloacetate again reformed when the citrate is subjected to citrate lyase, or when the citrate lyase is acted on the citrate. Okay, and this oxaloacetate will be converted to malate by the enzyme, that is a malate dehydrogenase. Malate dehydrogenase. So after this it will be converted to again pyruvate by the enzyme, that is a malate enzyme. So during this process there is a liberation of NADPH. So here we can see the liberation of NADPH by the utilization of NADP, and also there is a liberation of carbon dioxide. So this NADPH is requisite for the production of fatty acid from the acetyl-CoA. So in the first step there is a production of acetyl-CoA and NADPH from the mitochondria. So I am repeating once again the first stage of fatty acid biosynthesis. So during the first stage there is a formation of acetyl-CoA and NADPH. So how it will form? So here inside the mitochondria the pyruvate will be converted into acetyl-CoA and oxaloacetate. So acetyl-CoA will be formed by the action of pyruvate dehydrogenase on the pyruvate, and oxaloacetate will be formed by the action of pyruvate carboxylase on the pyruvate. So here the acetyl-CoA is impermeable, that means it is not able to cross the mitochondrial membrane, thereby it cannot able to enter into the cytosol. But we know that, so the cytosol of the cells has the enzyme machinery that are required for the biosynthesis of fatty acid. So the enzyme machinery is not available in the mitochondrial membrane or inside the mitochondria. For that reason, the acetyl-CoA should be available in the cytosol. But the acetyl-CoA cannot able to transfer through this membrane. For that reason, the acetyl-CoA will combine with the or will condense with the oxaloacetate by the action of enzyme, that is a citrate synthase. It will be converted to citrate. So this citrate has the capability to transfer this mitochondrial membrane and it will enter into the cytosol. And there the citrate will be again lysed or break down into acetyl-CoA and oxaloacetate. And this formed oxaloacetate is converted into malate by the action of malate dehydrogenase, and this malate is converted into pyruvate by the action of malate enzymes. So during this process there is a liberation of NADPH by the utilization of NADP and also liberation of the carbon dioxide. So these three are required or starting materials for the biosynthesis of fatty acids. Okay. So the advantage of this coupled transport of acetyl-CoA and NADPH is nothing but the transport of acetyl-CoA from mitochondria to the cytosol is coupled with the cytosomal production of NADPH. So there is a coupled transport is there. The advantage of this one is nothing but to produce the cytosomal NADPH and CO2. So which is highly advantageous for the cell for optimum synthesis of fatty acid. So this coupled transport of acetyl-CoA and NADPH is very much important. Okay. So next, second step of biosynthesis of the fatty acid is nothing but formation of malonyl-CoA. So the acetyl-CoA which is entered into the cytosol is carboxylated to malonyl-CoA by the enzyme acetyl-CoA carboxylase. So here the acetyl-CoA will be converted to malonyl-CoA by the action of acetyl-CoA carboxylase, and this step is the ATP dependent reaction, and it also requires the carbon dioxide, it also requires the biotin for the fixation of carbon dioxide. So the carbon dioxide and NADPH is utilized during the biosynthesis of fatty acid. So in the conversion of acetyl-CoA to malonyl-CoA, carbon dioxide is utilized. So that is fixed to the acetyl-CoA by the action of enzyme acetyl-CoA carboxylase, and also it is utilizing the biotin. So the biotin is required for the fixation of this carbon dioxide for this acetyl-CoA. Okay, and it is also a ATP dependent reaction. Okay, and the mechanism of action of the acetyl-CoA carboxylase is similar to that of the pyruvate carboxylase. So we already seen that pyruvate carboxylase is one of the enzyme that is converting the pyruvate to oxaloacetate. Okay, so like that there also the pyruvate carboxylase will play the same role in that situation. Okay, And here the acetyl CoA carboxylase. So the conversion of acetyl CoA to malonyl CoA, there is a one enzyme, that is acetyl CoA carboxylase is required, and this acetyl CoA carboxylase is the regulatory enzyme of fatty acid synthesis. So by inhibiting this acetyl CoA carboxylase, we can inhibit the fatty acid synthesis. So for that reason, it can be considered as a regulatory enzyme in the biosynthesis of fatty acids. Next, coming to the third stage of biosynthesis of the fatty acid, that is nothing but the reactions of fatty acid synthase. So there are 7 reactions occurred in this in the third stage of fatty acid biosynthesis. So let us see one by one. So the first reaction which is involved in this stage is nothing but the 2 carbon fragments of acetyl CoA is transferred to the acyl carrier protein of the fatty acid synthase. So here this is the fatty acid synthase protein, or it is also called as enzyme. So this one is called as the fatty acid synthase. This is the fatty acid synthase. So to this fatty acid synthase, so here 2 carbon fragments of the acetyl CoA is transferred. So that is the first reaction takes place. So that will be occurred by the action of enzyme, that is nothing but the acetyl CoA ACP transacylase. Acetyl CoA ACP. ACP means acyl carrier protein. Acyl carrier protein. The acetyl CoA ACP transacylase is the one of the enzyme that can transfer the 2 carbon fragments of acetyl CoA. So these are the 2 carbon fragments. These are the two carbon fragments of acetyl CoA. The 2 carbon fragments are transferred to the ACP of fatty acid synthase. So that is the acetyl carrier protein of the fatty acid synthase. So that acetyl unit is then transferred to transfer from ACP to the cysteine residue of the enzyme. So the ACP site will fall vacant. So when you see in this, so first the 2 carbon fragments of acetyl CoA will be transferred to the ACP. So this is the ACP, that is a acyl carrier protein. So to this protein, the 2 carbon fragments are transferred from acetyl CoA. Then these 2 carbon fragments are transferred to the cysteine residue of the fatty acid synthase. So this is the fatty acid synthase. It consisting of ACP and cysteine residue. 2 components are there in the fatty acid synthase. So first, the 2 carbon fragments of acetyl CoA will be transferred to the ACP portion of the fatty acid synthase, and again that 2 carbon fragments are transferred to the cysteine residue of the fatty acid synthase. Okay. And then again the ACP site is vacant, that is a free. Then in the second reaction, the enzyme malonyl CoA ACP transacylase. So in first reaction, acetyl CoA ACP transacylase, and in the second reaction, malonyl CoA ACP transacylase will transfer the malonate from the malonyl CoA. So this is the malonyl CoA. So from this malonyl CoA, there is a malonate will be transferred to the ACP of fatty acid synthase, to the ACP site of fatty acid synthase. So here we can observe that. So cysteine carbon 2 fragments are attached to the cysteine residue, and here to the ACP site from the malonyl CoA, malonate is transferred. Malonate means, so it is a C double bond O, CH2 COO minus, that is a malonate. So that is a fraction of the malonyl CoA. So that fraction is transferred to the ACP of the fatty acid synthase. So this will be done by the action of malonyl CoA ACP transacylase. And in the third step, the acetyl unit attached to the cysteine is transferred to the malonyl group. So here, so in the first step, what happened? Two fragments will transfer from ACP to the cysteine residue, and again this two fragments of carbon will be attached to the malonate, which is attached to the ACP residue, ACP site. Okay, so there is a bound to the ACP, the malonyl group which is bound to the ACP. To that ACP, again this two carbon fragments are again come and attached. So for that purpose, there is a during that process there is a loss of carbon dioxide. There is a COO minus will be removed in the form of carbon dioxide. Okay, and there is a and for that reason the carbon dioxide is never incorporated into the fatty acid carbon chain. Okay, so the decarboxylation is accompanied by the loss of free energy. So the carbon dioxide removal is accompanied by the loss of free energy, and which follows the process to forward. So this reaction is catalyzed by the beta keto acyl ACP synthase. Beta keto acyl ACP synthase. So that is a reaction take, and that is an enzyme which will forward the conversion of acyl malonyl enzyme to the 2-keto acyl ACP. So that is nothing but conversion of so that is nothing but the transfer of two carbon chains from the cysteine residue to the malonyl group which is bound to the ACP. Okay, and the fourth reaction, the beta keto acyl ACP reductase reduces the keto acyl group to hydroxy acyl group. So that reducing equivalents are supplied by the NADPH. So already in the first stage NADPH and carbon dioxide is produced. Carbon dioxide is utilized in the second stage, and NADPH is utilized in the fourth reaction of third stage. So that is for the conversion of conversion of keto acyl group of this beta keto acyl ACP to hydroxy acyl ACP. So for that reason, there is a reducing equivalents of are supplied by the NADPH. So that means, so here we can see this. So this is the beta keto acyl ACP. So here we can see so this molecule is nothing but beta keto acyl ACP, which is having the beta keto, and this will be converted into beta hydroxy molecule. That is a beta hydroxy acyl ACP will be produced. So this reaction is catalyzed by the beta keto acyl ACP transacylase. Beta keto acyl ACP reductase. So this enzyme will reduce the keto acyl group of the beta keto acyl ACP to hydroxyl group. So thereby it can form the beta hydroxy acyl ACP. And in the fifth step, the beta hydroxy acyl ACP undergoes dehydration. That means a molecule of water is eliminated and a double bond is introduced between the alpha and beta carbon. So here it is the beta hydroxy acyl ACP. So there is a hydroxyl group is there, and that hydroxyl group, due to the loss of one water molecule, so that will be removed, and there is a double bond is formed between the alpha and beta carbon. Okay, and in the sixth step, the second NADPH, So in the fourth step, one NADPH is utilized, and in the sixth step, the second NADPH dependent reduction is takes place. So by the utilization of NADPH, so that catalyzes the enol ACP reductase enzyme, and thereby there is a production of acyl ACP. There is a production of acyl ACP. So this is the acyl ACP is produced due to the action of enoyl ACP reductase. Enoyl ACP reductase. So that will reduce this trans beta enoyl ACP. Trans beta enoyl ACP is reduced to acyl ACP by the action of enoyl ACP reductase. So during this process, there is another NADPH is used. So due to this, the reduction will take place. So that will convert the trans beta enoyl ACP to the acyl ACP. That means the double bond which is present in the trans beta enoyl ACP will be removed, and it will be converted to acyl ACP. And this acyl ACP, so the carbon chain which is attached to the ACP is transferred to the cysteine residue. So that means here it is a butyl ACP is there in the acyl ACP. That butyl group is attached to the again cysteine residue. So thereby the ACP site is again vacant. So this steps from second step to second reaction to the sixth reaction will repeat for about 6 times. The reactions which has occurred from the second reaction to sixth reaction, that means the second NADPH utilization, that means conversion of trans enoyl ACP to acyl ACP, that is a sixth step. So up to that step, 6 times this process is repeated, and each time the fatty acid chain is lengthened by 2 carbon units. So that means, so what is the next step? So here 2 carbon atoms are come and attached to the again malonate group which is bound to the ACP, and it will be subjected to for the decarboxylation, and again reduction. So that means these steps are again repeated for about 6 times. For each cycle, 2 carbon elements, 2 carbons are attached to the cysteine group of the fatty acid synthase. Okay. And in the 7th step, so that means at the end of the 7th cycle of these reactions, the fatty acid synthase is complete and 16 carbon fully saturated fatty acid is formed. So that is namely called as the palmitate, and this palmitate is bound to the ACP that is occurred. And at the end of this 7th cycle, the 7th step is nothing but the enzyme palmityl thioesterase. So this is the one of the enzyme which can separate the bound palmitate which is bound to the ACP in the fatty acid synthase. Okay, so the palmitate is attached to the ACP site of fatty acid synthase. So that will be cleaved, or that will be separated by the enzyme that is nothing but palmitoyl thioesterase. So which will separate the palmitate from the fatty acid synthase by cleaving between the ACP and palmitate, and this completes the synthesis of palmitate. So this is the biosynthesis of palmitate. So that is one of the fatty acid. So the example given in this fatty acid biosynthesis is nothing but biosynthesis of palmitate. And coming to the summary of palmitate synthase, of the 16 carbons present in the palmitate, only 2 come from the acetyl CoA. So at the starting, 2 carbons are obtained from the acetyl CoA directly. The remaining 14 carbons are obtained from the malonyl CoA, so which in terms is produced by the acetyl CoA. So the malonyl CoA is produced from the acetyl-CoA in the cytosol. So the overall reaction of fatty acid palmitate synthase is summarized like this. So 8 acetyl-CoA plus 7 ATP and 14 NADPH plus 14 hydrogens will produce the palmitate, and 8 CoAs plus 7 ADPs plus 7 PA plus 6 H2Os. So this is the overall reaction of the biosynthesis of fatty acid. Okay, so that means, so at the starting stage we are utilizing the 2 carbons of acetyl-CoA and 7 cycles are occurred. 7 cycles. From each cycle, how many carbons are utilized? 2 carbons. So 7 multiplied by 2, that is a 14. 14 plus 2, 16 carbons. 14 plus 2, 16 carbons. So that is a carbon chain which is present in the palmitate. Okay, and we know that, so ATP is utilized in the second stage of this palmitate synthesis. So for the carbon dioxide fixation, so for that reason 1 ATP is required, and the third stage at 4th reaction and 6th reaction NADPHs are utilized for the reduction process, and 7 cycles are required. For each cycle 2 NADPHs are utilized, so that is 14 NADPHs are utilized, and 14 hydrogens are also utilized in this biosynthesis of palmitate. So finally we can get the palmitate and 8 CoAs, 7 ADPs and 7 PAs and 6 H2Os. Okay, so one water molecule is removed, and why it is 6 means, so at the 7th cycle we are not able to produce the 1 water molecule. That is the reason there is a 6 water molecules are produced. Okay. So this is about the biosynthesis of fatty acids. Next class we are discussing about the cholesterol. Thank you.